even throughout my biology studies, I was often confronted with schemes such as the one here that I just picked semi-randomly from a paper. And what you can see here is a bunch of proteins and arrows linking them. And this is what I'd like to call a mental model. So it is a sort of model which we try to make sense of what's going on. And what this mental model is asking us implicitly to do is to integrate in our heads what are all these processes doing over time and space. So that's quite complex. And I think that's one of the reasons why I felt compelled to move to mathematical modeling, to hope to put this in a more structured way that could help me make sense and understand what's going on in a cell. And I'm sure many of you can relate to that feeling. So what if we could really find, what if we knew what every piece does? And here I borrowed a slide from last SMB meeting from Stefan Ruland's presentation. What if we could know what every single part does? And using this very nice car metaphor, we could take all of this information and build the ultimate model of life, the universe, and everything. So this is, for those who don't know, it's a bit a reference to the Hitchhiker's Guide to the Galaxy, where the plot starts with an alien race that develops a supercomputer that should give the ultimate answer to life, the universe, and everything. The answer turns out to be the number 42. And then they realize, oh, we got an answer, but we don't really have a question. So I think that's kind of at the core also of the discussion today. If we know what every piece does, what then? What if we can simulate the cell perfectly? Would it still be useful? And I'm sure you're also familiar with the saying attributed to George Bock that all models are wrong, but some are useful. There's even an entire Wikipedia page discussing this very sentence, which is a bit remarkable. And to give us a bit more of food for thought, I thought about talking about levels of abstraction. Still going back to this car metaphor, so our perfect ultimate model would 100% be like a car, but we can also think of a toy model, which is what most of us work with our current level of technologies, little toy models, which still can help us understand something about the system. And that brings me to the last point, which is there's also different levels of understanding if you think about it. Because if I understand how to drive a car, I don't necessarily understand how to do repairs or how to build a car, how to make the parts that are used to build a car, how to get the materials, what the different atoms are doing in the parts that build the car, and so on. And for example, driving a car and to take it back to biology, it's a bit like doing cell culture, right? You understand the inputs and you choose the inputs such that you get a specific output. So yeah, that's about it. That's what I wanted to tell you and to start to get this discussion going. If we could simulate the cell perfectly, well, what's the use that we expect to get out of this model? Also in terms of levels of abstraction, different types of perfection that might exist and different levels of understanding we hope to get out of it. So that's about it.
So when this topic came up, Martin, I don't think he's here today. I didn't notice him earlier anyway. Martin's argument was that you could essentially, if you had a model of a complete cell, you could essentially, do computational experiments, basically go and poke at it, either mutate a gene or, give the cell a whole bunch of, some toxin or something like that in silico, and then develop a detailed understanding of the effects of some of these things from these in silico experiments, right? That was Martin's argument. um, I'm kind of curious, what folks think about that. I mean, I said a few things when this came up at the time. I'll say more later, I'm sure, but I mean, could we, would we actually get anything useful out of a model like that we can poke at and mutate and again expose toxins, whatever it is?
Well, in theory, it sounds nice, but in practice, you'd have to think about how long does it actually take to simulate it. Like the 3-page long PNAS paper about modeling bacteria and stuff. Like if an experiment's going to take like 2 hours in practice to actually do a treatment, and you can model that, but then it takes you a month to actually model the bacteria being exposed to this toxin for an hour, you may as well just do the experiment, right? It's not actually useful in practice because it would take longer to run the simulation than it would to actually just do it.
Yeah, but okay. So I'll try to be Martin for a minute. The counter argument would be if I do the experiment, I can see kind of phenotypic level responses, right? I mean, I can see that, maybe we engage some response pathway or other. I can, I can measure a few variables at a time, more and more as we go. But, an experiment where we measure 6 different things about a cell is still a really hard experiment, right? But if I have an in silico model, I can see everything. I can see exactly what the cell is doing and how it's doing it.
Okay, but it becomes a trade-off, like how long does it take, right? So if you can realistically do 5 or 6 of these experiments in a week, then you can measure, and you can measure 2 things per experiment, and you can do like 10 or 12 different measurements. I guess it pales in comparison to the number of things you can see in the cell if it's got like 20,000 proteins and you want to see exactly what each protein is doing. So yeah.
I want to bring up another issue, which is I don't really believe in any one perfect model of a cell, because I think that even if we're very careful in assembling such a model, lots of decisions have to get made at each step about how to represent things. And so we come up with something very complicated, which maybe we can understand, but who's to say that this is in fact the ultimate, the correct model? Besides which, cells are very diverse. So each cell will have its own set of parameters. How do we deal with that factor?
So Marcus is in the chat is writing, are we talking about a theoretically perfect knowledge-based model or something more akin what we can actually do?
So when the conversation started, it was definitely kind of, assume that, we're, way farther along in information technology and computer science than we are now. The experimentalists have gone and, done all the nasty detailed work to give us parameters and so on. And so you've got this, you've got a particular, cell that you have, so I mean, as Leah says, there's many different types of cells, but for the sake of argument, let's say that you pick, it's something reasonably simple but eukaryotic, just to make it interesting. Let's say you pick like Saccharomyces cerevisiae, and a particular strain of Saccharomyces cerevisiae. You've got everything in that cell model. Then you've got a computer that's better than what we do now, right? Than what we have now, right? There's been some great leap forward in technology that lets this be practical at all, right? So that was kind of the starting point for the discussion. But I mean, there's all these practical things, right? Obviously, and one of the things that Leah's brought up, which I think is very interesting, is you make decisions not just about what you put in the model, but how you model it, right? Dennis Salahub at the University of Calgary used to talk about this dream of a completely bottom-up kind of approach, which I mean, Dennis realized it wasn't entirely realistic, but it was an interesting idea where you would start with, kind of a quantum mechanical understanding of the chemical reactions that go on in a cell, and then, kind of somehow do some averaging or coarse graining or something. So you got a kinetic model, right? And then you'd introduce some transport, and, you'd basically, start from this very fundamental stuff and work your way up, right? Obviously doing a whole cell that way is not really realistic, but are there details in a cell where you need that level of understanding to get there? I don't know. I honestly don't know.
Yeah, related to Leia's comment, there's some studies suggesting like heterogeneity at the single cell level, right? Like a lot of the single cell genomics or transcriptomics are showing there's a lot of heterogeneity even within a single population of cells. So it makes it even more difficult to pick like a theoretical best model to really test, because how similar are they really? They seem to achieve the same thing, but with different, I guess, was it Dennis's, idea of like the quantum and molecular mechanics modeling? Like you can have all sorts of different combinations of that and get the same phenotype. So picking just one model, which you would then have to validate anyways. And if you're going to validate the model in every which way, right, to show that it's completely realistic, then you have to actually compare it to an experimental system. And at that point, you just have all the data anyways, right? So it might be useful after the fact, after it's validated.
Yeah, the validation would be a bugger.
That would take a long time.
But if it's perfect, then every time you go in and try to validate it, it'll match perfectly. So its success rate will be 100%, I guess, on every experiment.
Yeah, but you have to validate. You have to get through figure out that it's perfect first, and then
Sure,
it's accepted.
I'm just saying, if we have like 250 biological experiments, then they will all match because we're assuming it's perfect.
Yeah.
But I agree with your main point that it just matters how long it takes to use. If it's impractical, then it's pointless.
The other thing, though, I wonder is, would you actually need to redo? Actually, it's like a half-formed thought. Never mind.
So the cell is not exactly isolated. And in advanced studies, people talk about microenvironment. You talk cancer. So since quantum mechanics was mentioned, maybe we have a many-body problem, like in physics, which is known to be very difficult. And then the interactions are like, there is infimum of possibilities of interactions with other cells and the environment. So it's very important to understand what's happening in the cell while keeping in mind the big picture, which doesn't add to clarification here. The difficulties are, the list of difficulties is increasing.
I agree with Itamar. Actually, that was one of the first thing I thought of. I'm like, sure, you can model one cell, but as soon as you put two of them together, I'm not sure it will be easy to, like, it will be still useful, right, for me. So if you're able to create a single cell model, though I'm not sure a single cell model would be how useful it is for development, but you're definitely going to start needing more than one cell. And that's where the interactions will happen. And I think, yeah, if you can model a perfect cell and then figure out how to get them to interact and then make decisions, I think it could still be useful because we still don't understand a lot about that.
So again, let me play devil's advocate a little bit. Aren't all the other cells just boundary conditions?
No.
Well, that's a short answer.
So let's continue the devil's advocate. So if you have a perfect model of a cell, isn't cell interaction an emergent property?
Like the
In fact,
perfect
if it is a
model.
perfect model, it can also divide, right? It should be able to divide.
Oh, I see what you're saying. So you're saying you start, you have this model of a cell, and then it does all the things a cell does, including dividing, right? But then there's Turing's old observation that in biology, you never get form from nothing. There was always a kind of a there was always a pre-pattern, right? I mean, you don't get to a multicellular stage without having gone through a whole bunch of divisions, starting from an egg, which itself had some patterning, so on and so forth, right? So you you have to fake that somehow, right? Even kind of starting, even, well, I suppose, I mean, I guess if your model was, a fertilized ovum, if that was your starting point, you might be able to. That would be crazy though.
Yeah. And what about stochastic symmetry breaking? You could consider that that is not really a pre-pattern and just something randomly happens, like some, I guess, the outside perturbation that induces that symmetry breaking or would be.
I'm curious, do you know of any examples of actual stochastic symmetry breaking in biology? Because a lot of the examples I can think of where there's an apparent symmetry broken, there was actually a pre-existing axis or some other, something else that designated directions in some way.
I can't really think of
Organoid.
any
Organoid.
organoids. Yeah.
Organoid seems to do it. I mean, those are, if anybody's not familiar with them, so you have the cells in culture, or you get the in vivo model, but now they can create these little tiny organs in a dish, and organoids seem to show a lot of symmetry breaking, and it is a little bit stochastic from what I can gather. If somebody else had better, but that's from what I've read and seen.
Right. Yeah.
I'm not.
Sorry, go ahead.
So go ahead.
No, sorry, you go ahead, please.
I'm not super sure about this, but maybe some delta notch systems where you have like picking which cell is going to be delta expressing, which one's notch expressing, might be stochastic, but I'm not really sure because there's so many other cells in the system. Like, how would you really say that there wasn't a pre-pattern or something that was already going to kind of indicate that this cell is going to be notched? So yeah, anyways, Erica.
Yeah, I wanted to mention a similar system, which is wind reaggregation of hydras. So hydra is like a freshwater polyp, and what you can do is you can put it in a grinder and dissociate all the cells, and then you mix them together in a completely random fashion, and then they self-organize again. So you destroy whatever pre-pattern was there, and they still reassemble somehow.
But is it not possible some cells hold on to a particular fate and they act as organizers of some sort?
That of course might be that there is some unknown, but as far as the known organizer genes, they get completely scrambled and then they reform peaks.
Has actually anybody recently modeled this hydra reaggregation? Seems like a very cute problem. I mean, I know way back in the days of Gear and Meinhardt, they had some, simulations for pattern formation that might form the shapes. But does anyone know if people have recently studied this?
So I know one research group in Heidelberg that was looking into it, or had some people looking into it. It was the group of Anna Marciniak-Czoszka.
Marcos has a comment in the chat about stochasticity. I guess you know, I guess I would say, I mean, from my perspective, as somebody who came from the chemical sciences, stochasticity is inherent to every chemical process. You know, you've got, you know, you've got these random collisions in solution that cause, you know, diffusion to be this kind of nasty looking, you know, non-differentiable kind of process, right? And you have to you know, somehow find your reaction partners in solution. If you think about just the process of, you know, a transcription factor looking for its um, looking for a promoter to bind to, a specific promoter to bind to, that's very, very stochastic, right? I mean, you've got this, you know, this diffusion process in 3D. There's some suggestion that there's also some kind of sliding search. You know, the association dissociation processes from the DNA are highly, highly stochastic when modeled using any reasonable theory I know about. So there's actually quite a lot of stochasticity. And when we were talking about heterogeneity earlier, I think Serge brought up heterogeneity. I mean, that's where at least some of it comes from, is just this inherently stochastic kinetics, right? And that's the other challenge, right? If you were going to model a cell, right, and Serge kind of touched on that, is it would have to have some stochastic, um, you know, some stochasticity built into it, right? So the kind of, in fact, the little PNAS paper that started this discussion, Eaton and Netz, I think, um, they were contemplating a stochastic model, and you know, then you're not talking about like a trajectory. You're talking probably about running many, many replicates and getting some statistics, right? And then, so that just adds, in terms of computation, that makes it really bad.
Yeah, although initially it seemed like it took a shorter amount of time, but then if you consider, I didn't think about that, like if you consider the number of replicates that need to be done, then you may as well just multiply whatever single simulation you need to do, right? The amount of time it would take would need to be multiplied, as opposed to just doing the 1 or 2 molecular mechanics, like really ground up simulations. So it's actually a little bit worse than we're led to believe in that paper.
Yeah. But I mean, let's say for the sake of argument, I mean, at least in theory, quantum computers might be really good at, you know, at generating, you know, stochastic trajectories, right? At essentially solving stochastic Ds or some equivalent thereof, right? You know, and let's say this was reasonably efficient. The thing I've been thinking about, and I can't make up my mind one way or the other, I'd really love to hear what other people think, is I can't quite figure out if the thing wouldn't be so complicated that in the end I wouldn't get a whole lot more than I would from you know, from an experiment, which is to say I'd see a phenotypic response, but it'd be hard for me to figure out what to look at. You know, when I open the hood, so to speak, it'd be hard for me to figure out what to look at to figure out what happened.
Yeah, at that point, it's basically going back to the traditional biologist's problem of how do you know what a good question is to ask, and how do you know what to look for, basically. Yeah. And so a little bit of that is just like good old intuition from reading a lot.
Or by then we have enough information to know what parameters actually are useful, because once you do know enough about the cells, we should have already figured out also what parameters may or may not affect the system. I think we all do this. I don't know, at least from modeling perspective, that's one of the first things I would do, is figure out what parameters everybody thinks affect my cell or my system. And then I prove that a lot of parameters do not really affect it like we would have expected. And if you do have this perfect, let's assume this model is perfect, we also have some understanding what are the useful parameters for when you're looking at something, or at least I'd hope by then.
I think what I was thinking of more so is that if this model is so complicated, that it's difficult. You like as a single individual, you can't really actually understand it because it's too complicated. There's too many moving parts. You can't remember them all. Your brain, as soon as you try and remember one more parameter that might be impacting something, you shove it in your brain, and then another parameter you knew was useful just pops out. You know, it's too much. In that case, you kind of have to just rely on, like, is it still useful at that, or is it as useful as it could be? Because a lot of other models are simplifications that allow you to get at the key points of whatever is going on, right? So
I'm going to jump in now.
Sure.
I'd like to promote the idea of a hierarchy of models, where you have some that are toy models, perhaps, that have just kind of really basic universal principles that you think are at play. And then they don't give you a lot of detailed information. So you look at one specific case study and you get more detailed and build in things that are relevant to that particular example and keep going. So at each cycle, you have some insight, and then you can get deeper and deeper. Of course, when you get deeper, you get into more specialized questions or cells or types or phenomena. But what do people think about that idea?
I like
Leah,
Sorry,
I was going to say this. Sorry, Serge. I was going to say the same thing about 30 seconds before you started talking, Leah. And I think, anyways, I learned from you. And I want to ask one question, which is, could somebody post the link to the PNAS paper that we're talking about in the Zoom chat or the Slack channel, or just to point me to that because I missed it. And when the title of this meeting got sent around, I was reminded of a paper that's been really influential for me. It comes from Alex McGillner, June Allard, and Roy Woolman. The title is Cell Polarity, Quantitative Modeling as a Tool in Cell Biology. And this paper kind of lays out this idea of a hierarchy of models that Leah was talking about. I posted it in the Slack channel under random, but I can also put the link in the chat here. And instead of a hierarchy of models, they lay out sort of a plane of models, going from sort of focused to broad, to conceptual to mechanistic. And they argue that all these models have merit, and then the value is in what you can learn from them in different ways. Anyway, so this was just something that I thought of and I thought was a really nice way to sort of think about the models in the literature and to try and organize them in my brain. I've started to think about them in this mechanistic, conceptual, focused, broad. Might just be a way of organizing this conversation a little bit.
Thinking back to the point that was mentioned by Mark about the model being so complex that when you try to look under the hood, you just don't get anything out of it, any understanding. But yes, then Maria mentioned, well, but by building the model, you gain this understanding. So once you have the final model, you already accumulated the understanding. Whereas who is you, right? It's the people who made the model and the people who get something from the model. You know, once you have a model that is so advanced that you would call it perfect, you hope that it will be of use to other people. And then I thought to myself, well, then we would need these models to be somehow accessible and come with a very detailed manual that even non-experts, so sort of like a Wikipedia for the model, where we can, if we're interested in one particular aspect of that perfect cell for which we have a model, we can look for it and turn the knobs of the model such that we can look at that isolated sub-model, toy sub-model.
That's an interesting thought. What immediately kind of came to my mind is if we're advanced enough to come up with a model that's like perfect, would we not be advanced enough to come up with some kind of computer program to monitor it as well, so that we wouldn't have to kind of physically sort through everything ourselves, right? Like, would it be possible for it to just tell a computer like, hey, like if something's going on in this region, for example, right? let me know and I'll go look at it.
So an AI that helps you analyze the results of what the model's doing and why
Yeah.
it's doing it and how it's doing it.
Yeah.
There's a great example of that in the literature. In a paper by Alex Mogilner and okay, I'm blanking out on the name. I'll think of this in a minute, but it was a fantastic model for nuclei in Drosophila muscle cells and how they physically space each other out, how they form a coherent spatial order in the cells. And what they did is after considering a large number of potential models, they had machine learning analyze the results of thousands and millions of runs and figure out which of those models actually is consistent with the experiment. I'll find the paper in a minute and share it.
So while Leah's doing that, while Erica was talking, I was trying to think, is there another field of science where people have built, these massive models? Because if you think about what would have to happen to build a, a complete model of a cell, maybe the word perfect's the wrong word, but, a complete model that has, kind of all the parts of a cell implemented in silico somehow. It would be very much a multi-generational effort, right? It would be a thing where, somebody starts working on it and over decades, you have scientists, graduate students, working on this thing and adding to it and refining it and doing all the things we were talking about in terms of validation and so on and so forth, right? So you're talking about a massive, massive, massive effort that would go on for a long time. And the only analogy I can think of that's even close is global circulation models that are used in both kind of shorter term weather forecasting and longer term climate forecasting. And there's actually at least two classes of models there that are in fairly common use, but they tend to have this property that, I mean, a graduate student adds a thing about, cloud albedo, and then another person, adds a module that works out, kind of the effect of local cooling or warming on evaporation rates over open water, and somebody figures out how to do some of the PDE integration more efficiently, and so on and so forth. And so these things, some of these programs are, by now, some of these programs have heritages that go back 50 years. And how do people in that field kind of handle that? I think it's fair to say there's probably not one person who knows every detail of the insides of these programs, and yet they're still useful in the sense that they use them in some of the ways we were talking about, by running multiple trajectories with different boundary conditions, different initial conditions, and then doing some averaging and looking at kind of, what do, and often combining, and the other aspect we haven't talked about here is generally they don't use one model, they use 10 models that all have deep heritages and do things slightly differently, and they look at what are these, what do these things do on average, what do the outliers do, and then kind of make some statistical projections from that, right? And I mean, it's not just a big undertaking from the point of view of computational time, which we've talked about a few times. It's a big undertaking from the point of view of the commitment of an entire community to, kind of engage in this effort over a long, long, long period of time, right? And then having trust, and then you have, and I think really it would have to be, again, multiple models so that you weren't reliant on any one model and whatever its internal quirks were, right?
You just got the whole point of this whole group. No. I mean, in a more I guess dreamy version, why not? They do these whole human cell atlas. I mean, Gary's part of it. He's not here right now, but they have these whole different sets of groups that work together to build an atlas. I mean, and yeah, I guess most of us tend to work on our own, but why not? Why can't we somehow join forces and create this atlas of a theoretical model that may serve a good purpose? I mean, I don't believe that that's too far-fetched. It just, like you said, it requires effort. It requires people joining and knowing, that there's a greater good for this. But I think it could be possible.
I
I
guess
wonder.
the question in case of the atlas, right, it's essentially data collection. I feel like that's like a really different kind of consortium effort than what you were talking about here, which is essentially prediction. And I guess more generally, I have the idea of like a perfect cell or a complete cell simulation. I feel like it's an interesting discussion, I guess, in the philosophical sense, but I feel like you're kind of bringing it back to the data, and people mention like single cell genomics and well, kind of many data types. I guess I feel like a model which is testable and falsifiable, and thus can answer a question, is so far away from perfect. We're talking about removing 99.999% no matter what, right? Like, because I mean, just, molecular dynamics, biophysics, gene expression, transcription, fractal binding, epigenetics, and that's within the cell. And someone mentioned the microenvironment and external signaling, which is absolutely essential to controlling the internal state of the cell. So yes, I guess I'm just a bit pessimistic, but my question would be like, right, what for me, the really interesting question is, I think any model that I can think of a cell which is useful, it has to throw away 99% of the things that cell does. So the provocative question for me is like, what's the 2%? What can you couple, and to kind of rise above the kind of most basic useful models that we have now, without going so far as to say that we're going to do everything?
That depends on what you're going to ask, though, right? Depends on what your question is. For example, are you interested in how cells move around, or how they divide, or how they secrete stuff, or
yeah,
whatever answer, whatever question you're interested in will also tell you how much you need to throw out.
Yes, absolutely. Which I guess is why without questions, it's a little bit hard to, like, we're coming from very different places. Like, if your question is cell migration, then that's great. My question is probably cell fate decision making. How do you force a stem cell to choose between two possible commitment lineages? And that's controlled by gene expression, external signaling, et cetera, et cetera, et cetera. Yeah.
Well, I mean, there's some, I think I remember a study where they were looking for the essential genes in maybe Cerevisiae, and finding, like, of course, you have like the housekeeping genes of the cell that are essential, but you could throw out a lot of different genes, and as far as they could tell in the laboratory setting, not really affect the functioning of the yeast. So in that sense, you might make it might help to reduce your pessimism, I guess, is what I'm trying to get at. Like, if you can have experimental evidence that such and such gene has evolved to become basically a vestigial gene, like it's not really useful anymore, it doesn't really do anything as far as we can tell, then you could maybe remove that from your model and then make it a little bit less difficult to deal with.
But essentiality is a funny thing, right? Because you've got these, in some cases, you've got these genes that are conserved across, sometimes pretty much, all eukaryotes, that in lab studies, you can knock them out and nothing really happens. The cells seem to thrive. And yet, if it's that heavily conserved, it must have some purpose. Right. And so essentiality is always kind of with respect to an environment, right? And one suspects that a lot of these things are conserved because under certain conditions, they become absolutely essential to cell survival, right? But it's just that, if you give the cells a rich medium, you can get away with knocking out a lot of stuff, right? You can, if you just think about amino acid synthesis, right? I mean, you can knock out quite a few of the amino acid synthesis genes in most cells, and the cells are just fine as long as you give them a sufficiently, as long as you give them a medium that's got all the right amino acids in it, right? So yeah, I'm always kind of skeptical when people talk. I mean, it's always, when people talk about non-essential genes, yeah, there's occasionally some things that are well and truly non-essential, in the sense that a cell has multiple copies of a particular gene, and you knock one out, sure, nothing will happen. Or you have these cases where one protein can substitute for the function of another when you knock one out, and so on some level they're non-essential, right? You may run into haploinsufficiency effects at some point, but yeah, I always wonder about those. I mean, they're interesting. Those studies are interesting in that they tell you kind of, again, if you have a perfect environment, what do you really need? What's the minimum complement of genes that the cell can get away with? But
I don't think you even necessarily have to think of it as a perfect environment. It's more like, I mean, in some cases, there are genes that are involved in stress tolerance, and the stress only comes by once every, so often. Like every 100 years, there is a massive drought or something in a region, and therefore, so the gene that helps resist drought is important or conserved. So in that sense, it's not necessarily like a perfect environment so much as just not a really horrible one.
Yeah, there's lots of that stuff too, of course. Yeah.
Yeah, I guess I share Mark's concerns with essentiality, but like, let's say for sake of argument that we could do it and take a mammalian cell and reduce the number of genes we have to model from 20,000 to 100, say, which is a huge reduction. I mean, I'm still stuck because we can't model interactions between 3 genes dynamically correctly, right? So there's 3, we can't do that, and we've got 100 like essential genes. So I guess I'm afraid my pessimism holds.
I will want to talk to you about the 3 genes thing, because we can talk about the 3 gene, but I want to push you guys a little further out. What if, like right now, let's say I know that Adam and I are both interested in pushing and understanding how cells diversify and make these decisions to become a different type of cell, starting from a single stem cell, whatever it is. But I know also Mark works from about the internal part of the cell, and I can see each person. Some people work on motility. And what if we can put all of these different parts? And now I'm not just asking the question, how does a cell diversify? I could also ask the question, if I could also understand maybe I can incorporate what Leah's knowledge is. I think, when she showed us all the different models, and somehow put these 2 information together, and how much further can I push the system? I wasn't just thinking about like worrying about what to put in or out, but what if we can just combine our, like, I already know a lot of things about my system. I already know what works and doesn't work. I can tell you what parameters are make a difference and what don't in my theory. So what if we can incorporate all our different knowledge, and somehow we don't have to use all of it, but start showing how all these different parts and different types of models that we seem to be, specialized in work for the full cell, because we're all working on different aspects of the cell. I don't think about motility, and I definitely do not think about specific elements of, I don't know, molecules, but I do think about genes, and I think about how cells divide and how do they pass that information and how they make those decisions. But maybe motility will make a difference, because we do see it in development. And so this is sort of the level. Like, I don't really care if I'm working on yeast, and I get this actually a lot, not a lot, but majority of time from reviewers, because when I propose a theory, it's not about just a single species. But majority of the reviewers are like, pick a species and just show it in this one species. But if you're trying to think about these broad developmental questions, maybe we don't need to be specific whether it exists in yeast or not. Maybe conservation is enough. But I guess beyond just specifics for species, I want to say, how can we push our models to make them like stronger, and maybe have this like atlas, although I know Adam doesn't think it could exist. It is still data, right? We do have data, and each one of us, our model does generate some form of data, but that data can potentially be useful, like a robot or something.
Well, to some extent, we do rely on, let's say, pre-existing published models to make forward progress, right? So If, for example, if you're looking for a model for notch signaling, well, there's a bunch in the literature. You can go read them and, you know, select one to use. So in some sense we do that, but I think it would be great if we could join forces and in pairs or groups of 3 build on stuff that we each know. The slight challenges are time, space, and energy, and, you know, where you put your efforts, right? Because sometimes it's just easier to build on what you know, and it takes a lot longer to start talking to colleagues and, you know, how do I take a model in Mathematica and combine it with something else in a completely different platform. But yeah, I think it's a great possibility.
So to sum up how the discussion has been developing, I would say we're discussing how do we get to the perfect model, right? This is the road towards the perfect model and a maybe still somewhat distant future, but to get there, we need to join forces and do it like the climate scientists are doing, that they somehow manage to create a framework where they can share and collectively put together a more and more a model of ever increasing complexity. Also parallel versions of the model, maybe it's a bit like a software development thing where you have a main branch and then you have the side branches and people test 10 different side branches. So I think the spirit of the discussion has evolved into this direction right now. We have this dream and somewhere in the distance that we can all see. We'd like to have a perfect model, but how do we get there?
I have like a perpendicular point to make here. Thinking of like other areas where this discussion is relevant. I thought of protein folding, not exactly development, but there the physicists and chemists have been working on massive models to solve this protein folding problem. And it's been kind of stagnant. I mean, it's not my area, but that's my understanding. And people have been trying to build interpretable physics-based models, kind of like the geophysics atmospheric example. And records seem to have been smashed in protein folding by non-interpretable machine learning models. I think AlphaFold 2 this past year. I'd say non-interpretable, but not constructed in the same way we described. So I guess it depends how we define perfect or useful. But I think in the coming decades, the most useful early models for modeling the behavior of cells, for example, let's say our question, I think you need a very specific question. So let's say our question is, how do we make cell type A switch to cell type B? What genes do we have to switch? I think we will have non-interpretable models which excel at answering that question. And maybe they'll be perfect.
Coming back to something that Mark mentioned earlier, with examples of really big models, which I think Matthew touched on with the protein folding. Maybe astronomy would be a field that does that, where they're modeling the motion of many bodies through its space, or like, but then again, they're so far apart, you might be able to just treat them as more or less independent. So I'm not sure about that. But then maybe a little bit less conventional one would be graphics in video games, because as they've progressively gotten better and better, one company will hold on to, you know, their engine or whatever for simulating. They want to simulate water better. They want to simulate the motion of people better. And so with that, they probably end up with like one really massive piece of software, and it might not be, a lot of people put in work and built on top of that same thing. So that might also be another field that would be related to a lot of people working on one problem and one computational model like they do in climate sciences.
I want to just disagree quickly with a couple of things that were mentioned. I think our aim is not so much to get the perfect model, because I don't believe in such a thing, but to get to useful models that can give us some understanding of biology. The distinction with astronomy is, at least in astronomy, we have Newton's laws that we believe hold. So we have a set of physical principles. We just have so many particles and bodies that it's hard to compute what happens. But we have a set of fundamental principles that we think hold. That's not quite true in biology as yet. So that's an issue of, that's a difference between. And finally, with video games, well, yeah, of course, they become very complicated and require huge engines. But the idea there is just to present something that the human eye thinks is a real environment and has nothing to do with understanding necessarily the real environment. I mean, if you can make people believe that they're in swimming in water, that's good enough. You don't need to actually put in the fluid mechanics or whatever. But yeah, so right, I'll let it.
I think my point was less that, especially for the video games portion, not necessarily that they're trying to understand something, just that these are examples where a lot of people have worked together on constructing one model, right? So
Okay,
just to clarify.
Great.
And also to add, I agree though with Leia's idea of the hierarchical thing and getting to trying to describe the system or having models that are of sufficient abstraction, right? Like, you know, do we really need a molecular and quantum mechanical model of the cell, right? It depends on your question. So
depends on your question. Yep.
And at that point, it's the level of abstraction you need, right? So if we can do something like Maria said, where we can get lots of people and combine these different models at different levels of abstraction, then that's probably sufficient. The only real problem is how do you decide what your level of abstraction needs to be, right? And how do you find out the key rules that you need to use for that level of abstraction, which is, I guess, a lot of biology is finding those, you know, like cell behaviors, things that you can use as those key rules in your model.
Well, interestingly, oh, sorry, go ahead.
Just a one really quick point just to relate to that. What to put in your model, right? If we're talking about gene expression, and someone mentioned like cell type A transforms to cell type B, right? It's you want everything relevant in your model and no more. Just a reminder that it can be a lot harder than that because, right? Okay, I've got two cell types and one transforms into the other, and I'm going to ask which genes change during this transformation. Great, which genes change above a certain threshold? Maybe 2,000 genes change. But I don't want to put 2,000 genes in the model because I don't actually know which genes are driving those changes and which genes are changing as a result. So, right, I want to build a model that predicted the genes that change that lead to this cell type shift. But I'm still quite a long way away just because I have a very specific good question and I have all the components. I still don't have a network. I still don't have the drivers and the reactors. So it's, yes, it's still a little bit harder than that sometimes.
Yeah, my point was somewhat related, which is that one of the things that I thought was interesting when I read the Netz and Eaton paper was that they were focused very much on proteins, as if that's all there is in a cell, right? There were no, no metabolites, no lipids, and there's quite a lot going on in the cell beyond, beyond just what happens to genes and proteins, right? And so this question of drivers of kind of I guess the question that arises from Adam's comment is that there's not just these genes that you have to worry about, there's the connections between them, and they're made by often molecules that are not just direct gene products, right?
So I'm going to interrupt because it's 5 minutes over, and I know some people will have to go. And I think this was great. I hope everybody enjoyed it like I did. But our next meeting, do we want to continue on this topic or do we want to move to a different topic? And I like to have your opinion so we can set it up. Or if somebody else would like to present something from their lab, we haven't had that in a while. So if somebody would like to show us some of their work, that would be also wonderful. You don't have to do a lot. It just would be a 10-15-minute presentation and you can just ask questions. Maybe there is something that is bugging you, like an open question, and we can discuss it together. So that is also a possibility. Somebody would like to do that. No.
I wouldn't mind discussing our work. It's not perfect or even useful.
You're selling it. No, I would disagree actually. I think their work could be very useful. So yeah, that would be great, Matt.
Yeah, it touches on the question of, let's say you have a model of a single cell, what happens when they interact?
Okay, so I will leave that for you to sign up if you don't mind.
Yeah.
And I will move our topic, the other one, to another date because I still would need somebody to sign up for that one. That was great. And thank you all. This was actually fun. I don't know how, and thank you, Erica. I really appreciate you leading. It was great.
Yeah, I was about to say we should definitely thank Erica for catalyzing this.
Yeah, I love.
I did not choose the topic though; it was already there.
But that's
Somebody
okay.
else chose it.
You
but
stepped
you did
up,
great.
right?
Yeah, thank you. I appreciate it. So, is there any more comments? I put all the papers that I could that I saw in the chat in our papers spreadsheet. So it should be under today's date, and I'll probably try to also put them in the Slack channel. And if you guys want to continue this discussion, please feel free to do so on the Slack channel. And hopefully once this is also recorded, I will put this on the Slack channel. If you cannot access the Slack channel and you want a copy, just email me and then I'll send you a link. So I'll see you all in 2 weeks. Thank you very much.
Thanks.
Thank you.
Bye-bye.
Thank you, Erica. I really appreciate it. That was lovely. It was great.
Thanks. I'm glad it was a success.
No, I loved it. I love how you showed the car and everything. It was perfect. I appreciate it.
Actually, I didn't get to show it, but I actually do have.
Oh. No,
actually, I use it for presentations when talking to high school students. I did this a few times.